Um, so today we're going to talk about vaccines. As I'm sure no one is surprised, I had difficulty editing myself and have far too many slides that I won't be able to finish today, but that's okay. If you have questions, let me know. If that means I end up kind of switching around the order of some of this stuff, that's okay, because I really like talking about this stuff. So I have plenty of things that I can say. So most immunologists start thinking about vaccines when they start thinking about this experiment that I told you about on the first day of class. This is the first immunology experiment, or at least the first experiment that most people sort of cite as the first experiment done in immunology. This is also I'm telling you the sort of classic version, which even has like paintings about it and things like that. There are some really fabulous articles that have come out, I feel like recently, but I also feel like it was like 10 years ago. So I don't know exactly how many recently to me as an old person, that indicate that some parts of this classic story are actually kind of a lie. But I'll tell you at least the classic version of the story. Classic version of the story goes that this immunologist, the scientist, Edward Jenner, along with many people of his time period, was really scared of smallpox. It was a very deadly, very disfiguring disease. It caused a lot of issues. And in fact, you can read about kind of all sorts of historical events that were impacted by smallpox, which is another place I could geek out for a long time, but I won't. And he noticed, and people generally, when they recover from smallpox, have lifelong scars. And he noticed that milkmaids always had really pretty skin. So you can see some milkmaids in the painting. They have lovely skin, and he hypothesized maybe this is because they are getting infected with cowpox virus, or not that he knew what a virus was either, but maybe it was this cowpox infection was somehow giving them protection against smallpox. And so he actually did this first immunological experiment that was extremely unethical, where he took cowpox and purposely infected an 8-year-old boy named James Phipps. You can see that happening here. And then gave James Phipps smallpox. He did this again in other individuals. The second person he did it on was his own son. And in the end, he found that previous cowpox exposure, in fact, did protect you from smallpox disease. This is his paper that he wrote. He did the experiment in 1796 and published this in 1798. And we now know that this is sort of an example of something that immunologists think about a lot right now called cross-reactivity. This is an image from your textbook indicating cross-reactivity, in this case with antibodies. Because I have spent much of my time thinking about T cells, I would say that T cell cross-reactivity is also extremely important in this case, but I don't have a figure of that. So we'll just have to use the antibody one, but assume it's both antibodies and T cells. And so the virus that causes smallpox and the virus that causes cowpox are in the same virus group. So some of their proteins are similar, like the green one and the blue one, while some are different. So the ones they could all be antigens. You can see that when one is infected with cowpox, they can make antibodies, also T cells not shown, against all of the different antigens. And those antibodies can cross-react with the same epitope on this closely related virus. And so basically what the idea was, by giving this sort of mild virus, you were inducing an immune response that protected you against the highly lethal virus. And you might look at this and be like, okay, this is an old-timey experiment. This is, the first immunology experiment. But this experiment is actually incredibly important in like the history of the world. As I mentioned, smallpox was a disease that was pretty feared. There are all sorts of measures I could give you to tell you how bad it was. that's not like super relevant right now, so just take my word that it's really bad. People were really scared of it. And that virus has been eradicated from Earth. And so, because of the use of Jenner's vaccine and the distribution of that vaccine, and really it was largely that same vaccine around the world, that virus was actually declared eradicated on December 9th, 1979. So just about 45 years ago. You can see the World Health Organization declaration. The general idea, again, this is a story I could go down for a long time, is that smallpox virus now exists in 2 laboratories in the world in their laboratory freezers. Basically because at that time it was the US and the USSR. Neither trusted the other one to have the only one vial. So there are 2 vials, each in a freezer in a laboratory in those 2 places. Again, long story. I could go on about that too, but I'm not going to. And so we stopped getting the smallpox vaccine. We stopped giving it to people. In the US, they stopped giving it in the 1970s. And pretty much everywhere in the world, they stopped giving it by the early 80s. I am relatively certain that none of you have been immunized against smallpox. And none of you are probably particularly worried about getting smallpox because we actually got rid of this virus, all thanks to this Jenner experiment. Smallpox is not the only infectious disease that has been dramatically impacted by vaccines. So this is a static image. I'm going to show you the little YouTube video that's linked here in just a second. But basically, this is showing the number of people who died each year in the United States of particular infectious diseases before we had a vaccine against that infectious disease, and the number of people who died, and I believe this was 2012 data, of that same infectious disease in 2012. And so you may not be able to read the numbers super well, but the sort of orange fill is trying to indicate how many people before and after, and you can see that the orange fill goes away, and you can actually see a percent reduction here across the board. And so we have really dramatically impacted sort of human health and infectious disease. And there are lots of measures that show kind of how much we've impacted human life. There are some people who have done economic measures about things like return on investment of vaccines. In fact, we can see that immunizations have by far the highest return on every dollar invested, as opposed to things like, preschool or doing cardiovascular disease research or things like that. If we think about sort of contributions to public health and human life, like Team Immunology wins so hard because of these aspects on vaccines. And if we add to that some of the vaccines that have impacted agriculture and helped our food supply and things like that, the economic impact here is huge. So I want to show you this little video, partially because I adore this video. I know this is a shocking idea, but don't read the comments on this video because it's on YouTube, and never read the YouTube comments. What is going to happen in this video is you're going to see data from a bunch of different years. The year is in the upper right corner. And so you can see that this is going to start in 1912. The x-axis is going to tell you the number of cases of the infectious disease. So this is the number of people who are getting sick, not the number of people who are dying. Note that right now you can see that this x-axis goes to about 8,000, and this is per million, okay? Over time, this axis is going to change. So know that at the beginning we were at 8,000. Then you can see a whole bunch of different infectious diseases and the case numbers in the United States, the numbers at the end of the bar. The pink things have no vaccine, and as time goes on, we'll have vaccines. Really pay attention to when they're to green, because green is when a vaccine is available for some of these things. I was going to say on my previous slide about all of the, or my previous comment about all of the like amazing, like how much Team Immunology has done for the world in terms of inventing vaccines. There's actually there are a bunch of these vaccines that were all invented by the same guy. And so, in fact, sometimes people quote him as like the person who has saved the most lives in human history. I actually have like DVDs of documentaries about him and stuff. His name is Maurice Hillman. So, like, yay. So anyway, let's watch our little video because I like this video. So you can see measles in the early 1900s. It's way up there. The number of cases, vary per year. And now you can see, diphtheria, smallpox, they are moving their way down as we have vaccines against them. Measles stays up there. New things like whooping cough sort of take over. And we're starting to get a general decrease in infectious disease. Our x-axis has changed. Now we've got, whooping cough, the polio vaccine. We're definitely decreasing infectious disease. And now there's a measles vaccine. And you can see just all infectious disease over time goes down pretty dramatically. We have a new leader in chickenpox. We're in, the early 80s now. And we've, decreased our x-axis a ton. And you can see how many diseases that previously were big killers are just gone because of those vaccine recommendations. We get the chickenpox vaccine, and now we can see just this massive decrease in numbers. And here, we've basically gotten to below 80 as our axis for whooping cough. Now we're going to compare what was the highest number we ever saw for that particular infectious disease compared to what the number was in 2017. And you can see kind of how much of a decrease there is in all of these different infectious diseases, all largely due to vaccines. Clearly, it's not 100% because you can see there was that decrease happening pre-vaccine, but a lot of this, particularly you can watch sort of that precipitous drop that happens with each of these following vaccine administration. And so vaccines are an incredibly, incredibly important part of our public health arsenal. Again, yay. So we need to think a little bit, of course, as immunologists about how vaccines are working. And they are really based on the immunological phenomenon that I show you here about secondary or memory immune responses. And so we've talked about secondary or memory immune responses when we talked about immune response kinetics, where we have a situation where one could be infected with a particular virus. When we're going to have our adaptive immune response, we'll see that there'll be that little gap in time. Eventually, we'll get an adaptive immune response. But while the virus is still reproducing a lot, we're going to feel sick. Some of that may be the immune response, some of that may be the virus. And that once we have a nice high adaptive immune response, we're going to get rid of that virus. Once the virus is gone from our body, we're going to start feeling better. And we're going to be living happily ever after. Until potentially some point where we get infected with that same virus a second time. If we get infected with that same virus a second time, then we get this memory response where we make this much larger adaptive response. The infection is happening, will probably be much shorter because our immune response will sort of block that virus right away. It will also probably be a reduced amount of virus. And so it's very likely that this person may not ever know that they were sick, may not feel ill. as opposed to the other things, like the antibiotics, the antivirals, are the things you do when the bad stuff has already happened, like go to the ICU. So like, there are two different ways of helping you be in good shape. And so that's one of these big parts of how vaccines are protecting us, is that they are giving us what I would call here individual immunity. So one reason why I get vaccines is that I myself want to be protected. Just like I myself wanted some donuts today, so I went and got you guys donuts. But there's also a second benefit. Just like my buying donuts benefits you guys, my getting a vaccine also benefits other people. I totally did not plan that with the donut thing, but now I'm so glad I did it. Because we have this additional set of effects, which are population level effects, in addition to the individual effects. And the idea with the population level effects is that basically what we can do is that if I am vaccinated, even if I get infected, say, that infection is going to be more mild than it would have been otherwise. I have less virus in me than I would have otherwise. And as a result, I'm not going to spread, the probability of me spreading it to you is less. So in fact, my getting vaccinated stops spread throughout the population and stops the probability of an epidemic. So you guys are protected because I got vaccinated. And when we think about this at a whole population level, we find that there's a bunch of math actually involved in this, in finding out kind of what percent of the population needs to have immunity before we actually can basically stop spread throughout the population. And so I'm going to say more about this, but here's kind of an example. So if we have a situation where everyone in the population is susceptible and somebody gets infected, then that infection can kind of spread through this whole thing like wildfire. So everybody is going to get infected at this situation. If we alternatively have a situation where some people in our population are immune, then that's going to stop spread. This person is not vaccinated, they're not immune, but they're unlikely to get infected because we can't have like a spread of infection to them. If we block infection in all the people around them, then they're okay. And so this is sort of the idea that we're talking about here. And the exact percentages will vary a bit. And so I'm going to show you, and so again, sort of having a specific percentage or level or threshold of immunity among the members of the population is really important. And when we see big differences in what happens in the population above and below that particular threshold. The threshold is different for each different virus, so what percent needs to be immune will vary for each different microbe, but now you're hearing how much I like viruses because I was using viruses as my example. And so you can see that here. Again, I will show you a couple of ideas of that, and that's something that you were looking at in the assignment that is due today, where you were using the simulator to actually look at some of the percentages that were needed to protect individuals. So I guess a couple things. One of them, there's one thing that I'm going to say right now that is 0% related to the news at all. I can't imagine any reason why the sentences that I'm going to say right now are related to the news. But the one thing to note here is that this slide does not say vaccinated. It says immune. And so technically, any way of people getting immune counts in the math that we're going to see. Basically, as long as you're immune, you get this immune response from somewhere, you're okay. And so some people argue that, well, yeah, this is actually data that says everybody should get infected as much as possible. We should get the immunity from infections instead of getting the immunity from vaccines. There are arguments to that effect. What's the problem with that argument? Yeah.
If you're trying to prevent the disease, why would your solution be everyone get the disease? Or infection, sorry.
Right. Yes. Yeah.
You have to survive the infection?
You have to survive the infection. And so the vaccine is not a thing that is potentially going to kill you. The infection is. So yes, it's true. If everyone got infected, that would also work as much as everyone getting vaccinated. But a bunch of people would die if everyone got infected, and they wouldn't die if everyone got vaccinated. Again, not related to the news at all. A couple of different ways that I want to show this to you. Just so you know, if you like any of the things I'm going to show you, these are the links. If you want them for, convincing people who you might ever talk to vaccines about ever in your life. I've got some really good links throughout this, and I got some good vaccine links. But there are two of these that are particularly good. All right, so this is going to show you the impact of the percent of people who are immune on spread of an infectious disease in the population. So we've got these different rectangles, which are different populations. And there's going to be sort of a darker green color, which is going to be the people who are susceptible, or as this says, vulnerable. So in this rectangle, 100% are susceptible. Here, 75% are susceptible. Here, 50% are susceptible. And we're going to see then at the top, you can also see how many people are immune. And then what we're going to do is we're going to watch the infection spread through the population, and the infection will be red dots. If someone is immune, either because they had been vaccinated previously, before this whole scenario, or because they recovered, they will become a light green dot. And so what's going to happen in this is that they're all going to start kind of with red dots and red spread at the same time in each of the rectangles. And what you want to look at is how quickly does everything spread through the rectangle? How many people in the end get infected? And notice whether, is the 75% really like 3/4 of 100? Is 50 really like half? Because what you tend to notice with this is there's a threshold number, as I kind of already told you, where it's like below that number, it makes a difference, but not a huge difference, but above that number, it's like done. And that's going to be really obvious here, as well as in the next little video I'm going to show you. Oh, I guess vulnerable wasn't dark green, it was brown, whatever. And so you can see that sort of once you hit some level of thresholds of people who are immune, we really see no spread in the population. But we were up at this 95%, literally the one person got infected, and that was it. So the idea is that we as a population care a lot about what the level of immunity is amongst our population to kind of help protect us all. And the video is going to keep going. I'm not going to go all the way through the rest of the video, but it's going to show it again with a different infectious disease that has some different properties. And so here you can see that the threshold is in a different place because of some of the different properties of the different infectious disease. And so there are kind of a lot of specific things that will change exactly what those numbers look like. The other thing that I really like about this is this one. So measles is one I'm going to refer back to a lot. People think about measles a whole bunch. And this is a fantastic simulator. One of the things that you will notice in the homework that you were doing was that there were a bunch of numbers that matter. And you can kind of have seen that in the previous video as well, that numbers about the particular microbe influence exactly what this pattern looks like. Some other things that influence what this pattern looks like include some things about the population. And so this, including things like population density. You know, if you live off in the woods by yourself, you're not going to spread viruses as quickly as if everyone lives close together. Like this. So this actually uses real census data about things like population density and populations in places to show you a model of what data would look like. And so you can actually put in different places. The closest place to where we live is Newark, so I'm going to pick Newark here. And so first I'm going to show you this one on the left. No. And I've been doing this for I don't know how many years, and I think I mess that up every time. So this is Essex County, New Jersey. Drew is sort of over here. And the red dots are going to be infected people, blue dots are going to be recovered people. And you can see what day we're at of the simulation here. So we're going to watch what would happen in a measles outbreak in Essex County if the percent of people in Essex County that were vaccinated was 80%. And so this is what that would look like. And 80% seems pretty high, right? That seems pretty good. So you can see in 238 days, kind of what that one measles case in Essex County would turn into with 80% vaccine coverage. Not great. I don't particularly want measles. So now I'm going to show you the exact same scenario. The only thing that's going to be different, it's going to be 238 days again, it's going to be Essex County again, everything's going to look the same, is that we're going to change what percent of the population is vaccinated. And so we're going to go from, instead of 80%, which again, sounds pretty good, right, to 95. So here we are, and I think it's done. It's just going to keep going to 238, so that you get the equivalent number of days. So you can see that 15% difference gives us much more than a 15% difference in what we actually see, because it's about being on either side of that threshold of numbers of infections. So sometimes when people look at data like this, they say, awesome, I totally figured it out, got it all figured out. I don't need to get vaccinated. I just need to make all my friends get vaccinated. And then I'm good. I just make everybody else get vaccinated, and then we're fine. So one thing I want you to realize is no vaccine is 100% effective. There are situations where people get vaccinated and the vaccine doesn't protect them or doesn't work. And so this percentage that we need is actually in what percent of people do we need the vaccine to work to get these effects to happen. And you can note, for example, when 80% of the population is immunized with measles, only 76% is immune. So we don't have a lot of room for error in people not getting vaccinated here, especially with things like measles. The other piece of this is there are people who cannot be vaccinated, either because they are allergic to some vaccine component, or because they are immunocompromised, either because they're on chemotherapy, they may be on some immunosuppressive medication to treat transplantation, to treat autoimmunity, to treat one of many things. The number of immunosuppressed people in our population has actually been increasing quite a bit. And so many of those people can't be vaccinated. As a result, the rest of us really have to be. in order to get these numbers where they need to be. And so I was telling the story to someone the other day about a family member of mine who suddenly realized out of the blue that they were immunocompromised. And I was like, yes, that you are correct. Um, and they're like, wait, does that mean that everyone else's vaccines protect me? And I was like, yes, that is how this works. And they were like, I gotta tell all my friends. So, know that when you're getting vaccinated, it's not just about you. It's about, you can imagine whoever in your life you want who might be more vulnerable, and the fact that you are protecting them. So I have spent a lot of my research career thinking about vaccines and vaccine generation. And there are a bunch of things that we can say about vaccines and how we generate vaccines. Successful vaccination really depends on inducing the right immune response. Once upon a time, historically, that was done by trial and error. Maurice Hillman did it by trial and error. Now we know enough immunology that we can actually do a lot of this design on purpose. And so the point that I want to make, that I'm making here, is that, oftentimes people think about, well, we're going to give you a vaccine to get an immune response. But it turns out just an immune response isn't enough. You actually need to get the right kind of immune response. So maybe if you get any old antibodies, it doesn't work. But if you get neutralizing antibodies, it does. Maybe you have to get a particular kind of T cell to respond or something like that. And so knowing about these types of immune responses and how to induce them is really key for making a vaccine work. The other big thing that I think is is a huge debate among immunologists, vaccinologists, public health professionals, anyone who thinks about vaccines, is really going back to something that's shown on this slide. If I go back to Maurice Hillman making all the vaccines in the 1960s, he made a bunch of vaccines. And, the idea was that he was getting this amazing protection. Yay. If you asked him what he was protecting against, he would probably have said he was protecting against infection. Because he didn't really spend much time thinking about the difference between infection and disease. He didn't have a way to measure whether somebody got a teensy tiny amount of virus for a day. The only thing he could measure is if they got sick. And so if they didn't get sick, then they must have been protected from everything. And that has really led to this idea in the public that you get a vaccine, it's like you have a shield that protects you against the infection forever. You're protected from that virus totally. It is never going to set foot in your body. Not that viruses have feet, but whatever. But what we know from basic immunology is that this is actually not a protection from infection, it's a protection from disease. You may still get a small amount of replication of the virus. So, for example, so this question of, do vaccines protect against infection or do vaccines protect against disease? And do random people on the street know that? And what do they think is success? What do they think is a successful vaccine? Is a vaccine successful if it keeps you from getting severe disease, but you still get mild disease? Is a vaccine successful if it keeps you from getting disease, but you still got infected and have no symptoms? Or is a vaccine only successful if it keeps you from getting infected? And if the virus can get in at all, the vaccine was a failure. That's something that people have actually been arguing about in the past 5 years, because it turns out that we never told the public this. And the public's like, wait, I still got infected after I got my vaccine. Your vaccine failed. You lied. All of immunology is a lie. When really it's like, no, it's actually that we never told you we were going to protect against infection. We told you we were going to protect against disease. Maybe we weren't actually clear on telling you that, but that's really what the idea is. back in the day, nobody made those distinctions because we didn't have a way to measure that little tiny bit of infection. We only could measure, did you go to the hospital or not? Now we have super sensitive PCRs and rapid tests that you buy at CVS and all sorts of things to detect little tiny bits of infection. And this has largely been the first big epidemic where that's ever been easily like something the public can figure out for themselves pretty easily. And so that's been one of the reasons why there's been a ton of sort of controversy on this. And one of the things that I sometimes get frustrated about when I'm reading sort of media accounts of different things related to what's going on with vaccines is people will talk about this vaccine induces a response or not, or it works or not. Not it works at preventing disease. It works at preventing severe disease. It works at preventing infection. It works at making antibodies. It works at making neutralizing antibodies. And what I want you to notice from all this is actually that's what's really important, is having to make that specification. It's not about I got an immune response or I didn't. Sometimes people write random emails to me pretty frequently, and I will get emails from random strangers who will ask me, what is the thing I can do to have the strongest immune system possible? And I kind of want to be like, well, I don't know if you want a strong immune system, because then maybe you have autoimmunity. Like, it's not just yes or no. There's all these variations and gradations and kinds and things. That's generally not what they want to hear. But no, that's a big part of this, is that I think that people have often thought about vaccines as an on-off switch and haven't thought about all of these levels of detail. So these are actually the data from the Pfizer COVID vaccine to kind of sort of put this into context. So this is actually how we figure out that a vaccine is effective. There were two groups of people. One group got the Pfizer mRNA vaccine. That group included 21,669 people. And a second group who got placebo, 21,686 people. And then those people were monitored to just see if they got COVID, to see if they got symptoms. So again, this is when this vaccine was made, it was about, does it protect you from symptoms? They never did. In this whole paper, you can read this whole paper, they don't actually look to see if it prevents against infection. They don't look to see if people got PCR positive but didn't get sick. They looked, in fact, you have to, when you read this paper, there's a whole section where they have to be very, very, very specific about how they defined what COVID was. They had a list of symptoms, and a person had to have at least two things on the list. So if you only had one symptom, that you didn't count as having COVID. So it was basic. So they were counting you as yes having COVID, no not having COVID, but they had to have a really strict definition. And then so basically, like I said, they just gave people this vaccine and waited. It was a 2-dose vaccine at the beginning, so really kind of the important stuff is starting around day 21, which is when that second dose is given. The red line is how many people with the vaccine got COVID. The blue line is how many people who didn't get the vaccine got COVID. And so you can see over time, people with the placebo just kept getting COVID, and the people who got vaccinated really didn't. It's not that 0 of them got it. There are a couple who got it after the vaccine. This line does go up a little bit after the end of vaccination. So it's not like it protects everybody, and if we actually look at the data, that we can see that. And it's not like didn't even look to see if it protected against infection. We were specifically looking, does it protect you against disease? And then they can do a bunch of specific calculations to find out how effective the vaccine was. And they look at that based on like what percent of people who got the vaccine got COVID versus what percent of the people who got placebo got COVID. And when they do that calculation, they come up with 95% vaccine efficacy. So that doesn't mean that like 5% somewhere got infected or something. There's actually quite a lot of calculation off of these data that get to 95% vaccine efficacy. So what I want you to notice from this is, it's not about protect from total infection. It's about protect from disease. And this is a big, very large, very complex study. It's not about did one person get, it's not about an anecdote of, well, I got the vaccine and then this happened to me, so we're going to throw it all out. This is looking at over 40,000 people. Also, not in any way relevant to the news. One thing to note, and one thing that actually makes some of this challenging in the vaccine development world, is here you note that we have vaccine compared to placebo. If I today discovered a new COVID vaccine, so let's imagine I today was like, no, I got something better than mRNA. I got a donut vaccine. Okay? And I want to do I want to do a trial of my donut vaccine. I actually could not do a trial like this. I could not trial my vaccine against placebo, because it is actually unethical to not vaccinate someone now that we know this vaccine works. My placebo group, I would be actually withholding good treatment from them by giving them a placebo. And that is not an ethical thing to do.
So it's only if there's no other existing
vaccine. So you can only do a placebo trial if there's no existing vaccine. You actually, if there is an existing vaccine, have to do something called a superiority trial and show whether your vaccine is superior to or as good as the one that's out there. And in some cases, if a vaccine's really good, there's not much room above it to prove above it. And so that's one thing that I will just point out as well. Only because, again, definitely not because anyone in the news has said that all current vaccines are illegal because they haven't been tested against placebo. Because we currently have I think it's a third or fourth generation measles vaccine, which was tested against the previous generation instead of against placebo. And again, not that there's anyone in the news who currently says that vaccine is actually illegal because it wasn't tested against placebo, but that's okay. Sorry, Brian or Jonah, do you have a question?
No, it got answered.
Okay, did you have a question, Brian?
Kind of. What are the ethics of when you're testing a new vaccine, where there's already one that is reasonably good? How do you determine the ethics of
So you have to do a superiority trial. So you have to see, do you get this kind of effect over the previously existing one? Sometimes, and so one great example of this, and that can be hard to do. There are a bunch of reasons why some vaccine trials are really hard to do. One great example of this is the smallpox vaccine / the mpox vaccine. The mpox vaccine is really just a new generation smallpox vaccine. It's really hard to do an efficacy trial against sort of the old Jenner vaccine. But in slides that I surely will not get to because of time, the old Jenner vaccine had some pretty rough side effects. And so what's actually they were able to do was show that the mpox vaccine was as good or better, sort of unclear, but has way fewer side effects. And so there's a little bit of can you show it has some other benefit while maintaining the same efficacy? But one kind of other thing to point out related to that is that in order to do this COVID vaccine trial, these experimenters had to know that some people were going to get COVID. The idea was they gave people the vaccine and then they waited to see how many of them got COVID. If I gave you all a smallpox vaccine right now and waited to see how many of you got smallpox, spoiler alert, nobody's getting smallpox. And nobody in the placebo group's getting smallpox, and nobody in the vaccine group's getting smallpox. So you actually have to have cases, and you actually have to mathematically know about how many cases you expect in order to do things like statistical power calculations of how many people you have to have in your study. There's a lot of that kind of stuff. That means that, say, doing a vaccine trial for a virus that, so one of the big places this was an issue was with Ebola, because Ebola doesn't lead to outbreaks that are really easily predictable. And so we couldn't vaccinate a population and check how many of them got Ebola, because we didn't know how many, that maybe nobody was going to get Ebola that year. So the whole thing was going to fail. And so there are actually some other issues in this that are related to what you know about the population and what you know about population infection rates that also influence some ways you do vaccine trials. Yes.
So for vaccines in general, are they mostly all mRNA?
No. So I'll come to that I'll speed through some stuff so I can make sure to kind of hit that a bit. Also, there are a bunch of other rules that are required before a vaccine can be approved. One part of this you can see is about you have to get protective immunity in the population. And so we have to think about, well, which antigen? Do we need antibodies? Do we need T cells? Do we need neutralizing antibodies or any other kind of antibodies? Ideally, you want the protection to be long-lasting. You can look at the data that's out there right now with COVID and find out how many people got one COVID vaccine, how many people got two, how many people got three. If your vaccine actually has to have multiple doses, if it doesn't necessarily last as long as one might like, it's not as many people are going to get it. If you can get a vaccine that only has to be given once in the whole life, you're going to be able to get to more people. The World Health Organization prefers vaccines that will cost less than $1 per dose for manufacture. You ideally want to think about that vaccine being genetically stable, so you don't want a vaccine that is going to genetically change once you put it in people. You need to think about how you store that vaccine. And so this again came up during COVID. The first round version of the mRNA vaccine, at the beginning they thought it had to be stored at -80°C, which many doctors' offices and pharmacies didn't have. Though like many labs have it, and so labs could do that, but like then how the heck do you get it to a person? That becomes a bigger issue when you're thinking about how do you send it around the world, when you may have unreliable electricity to refrigerate in different places, things like that. How do you deliver it? A lot of people are afraid of needles. They don't want needles. Sometimes some types of delivery take a person who's been trained a lot. Ideally, if you want to go around the world, you may not have enough trainers. You may not have enough needles. So how do you do delivery? So that's a big question. Everybody always asks, like, why can't you just make my vaccines a pill? Why can't I just ingest it? That would make everything better. Then I'm not afraid of needles. Remember that I've mentioned a couple times this idea that your GI immune system is different than immune systems in the other parts of your body because you don't want to make an immune response to food. You actually, generally, if you encounter an antigen by eating it, you end up with tolerance. So if I gave you all your vaccines orally, you would then become tolerant to measles and tolerant to mumps and tolerant to influenza, which would be like worse than not having any immunity. So learning like why is it that that immune system gives you tolerance, and how does that happen, and can we mess with it, would be really key. There's also, of course, this huge question of safety. And the idea, of course, is that the vaccine needs to not cause disease and cause minimal side effects. Part of the issue with this is what is a minimal side effect in one person, or for what one person says is minimal may not be minimal to somebody else. Like different people are going to call even the exact same side effect different. And the specific side effect will vary a bit from person to person. Many of the side effects are rare. You can see in the Pfizer vaccine trial I showed you, they looked in 40,000 people, which was like a crazy huge vaccine trial. If something bad happens to somebody at a rate of 1 in a million, you're not going to see that in 40,000 people. You are going to see it when you go to 8 billion people in the world. Some people are like, this vaccine is not safe, it gives you a fever. And so like, what does the safety mean? There are certain effects that happen if I give you a syringe full of saline. And so, what is that bar is, again, something that is very much debated. Again, in the list of things that I am not going to have much time to talk about, there was a very famous vaccine complication case where a vaccine was manufactured incorrectly by a pharmaceutical company. This was in the 1950s. For those, if you guys think about lab work and things, in preparing the vaccine, the lab worker went into the lab and picked up the wrong filter. They used the wrong filter. It was really like they went into the room and then picked up the filter to use for the vaccine lot and picked up the wrong one. They used the wrong filter. The vaccine ended up killing some people, paralyzing some people, causing a lot of issues. As a result, we actually have like a whole vaccine court and a whole vaccine system. Things like that so do not happen anymore. And we actually have probably one of the best reporting systems that's out there. You may have heard early in the COVID vaccine some discussion of some heart issues, some myocarditis in certain patients. That came through the reporting system, with the idea that basically the CDC has a system where if you have any kind of side effect that you're worried about, you can actually report it yourself on the CDC website. And if they start to see a pattern, not just like, oh, that one guy, that guy, you know him, said that his arm hurt. But if they start to see, oh, there's a pattern that like a bunch of people are all seeing the same thing, they will study it and they will like add it to the list, and that may change approvals and things like that. That really does happen. It's called the Vaccine Adverse Event Reporting System, VAERS. Be very careful if you ever look at the VAERS data. And the reason why I will say that, again, no way related to anything in the world. Sometimes people will be like, well, if I go on the VAERS website, like I can find a person who reported blah, blah, blah happened to them. So this vaccine must be bad. It's not monitored. It's literally, you could go on it right now and write whatever you want. And so no, it really is like that. And so some people are like, oh, well, they're just hiding stuff. No, they really aren't. You can look. And to make the point of, yes, you could write anything, so you take anything you see there a little bit with a grain of salt, but also no, they're not hiding anything. A vaccine researcher, once for fun, wrote a report that a vaccine turned him into the Incredible Hulk. And it took 2 years of arguing with the CDC to take that off. Because they're like, no, it was a report that was written, and the way our rules are done, if a report is written, it must stay. And so there was a joke report about a vaccine turning someone into the Incredible Hulk that was up for 2 years. So, like, be ready for that. Yes.
I just wanted to know of an example of an oral vaccine, because I can't think of any. And is it more, or wouldn't it be less effective than that?
So I can think of 3 examples. So there used to be a polio vaccine, or there is one polio vaccine that's given to some kids orally. There's a rotavirus vaccine that's given to some kids orally, and there is a typhoid vaccine that can be given orally. There are 2 versions of the typhoid vaccine. One thing that all 3 of those pathogens have in common is that they specifically reproduce in the intestine. And so actually messing with the immune response in the intestine can be important. But there's also a typhoid vaccine that's a shot, so it doesn't have to be. All 3 of those were really trial and error.
So it's more like what it's like. You could have arguably made COVID oral, but it's not. That's not the type of infection it induces.
So I'm gonna skip ahead in a second, but no, it's okay. Before I skip ahead, I will just say that there are people who are trying to actually make vaccines that you shoot up your nose. Because so many, and COVID is one of the ones that they have as an example for that, with the idea of, well, we want to have you have a really good nose immune response. So this is a study looking at nasal delivery. There are a bunch of issues with nasal delivery, but people are trying to figure it out. It just hasn't been figured out yet. I will tell you, having done that in guinea pigs, one of the problems in doing mice or guinea pigs or any animal models and trying to test your nasal vaccine is they have an amazing sneeze reflex. The number of times I've been sneezed on by a guinea pig after putting the vaccine up their nose. So, the sort of I'll spend the rest of the time addressing Juliana's question, although again there are a million things I could talk about. So there are a bunch of different vaccine types that are out there. I will kind of hit highlights of some of them. This shows all of the different vaccine types that were done, that had research done on them for COVID. I'm obviously not going to be able to go into all of this in like crazy good detail, but at the top there's a really nice link that goes through how the COVID vaccine, particularly the mRNA vaccines, but all of the versions of COVID vaccines work from New York Times. And Pfizer has a crazy good website that goes through how every vaccine type works, except that they, on mRNA vaccines, they only talk about the Pfizer one and not the Moderna one, even though they both work the same way. They just don't write their competitor's name on the website. But it's the same, but both of those websites are really good. So the original idea, the first thing that people did was they said, well, what we could do is we can take the virus or the microbe and we can weaken it. We can make it so we are giving it to you in a weakened form, so it can't cause disease, but it could still induce an immune response. That's known as a live attenuated vaccine. And so your measles vaccine, your mumps vaccine, your rubella vaccine, all are live attenuated. A bunch of vaccines that you get are live attenuated. So that was kind of the first idea people had. These vaccines are great because they give you crazy strong immune responses. They give you like the best immune responses, but they also have the most safety concerns. They could mutate and cause some issues. They can cause issues if someone's immunocompromised. They cause disease in some people. So it's a live, reproducing form of the pathogen, but it's weakened. But these are sort of the least safe, but they give you the best immune response. Some ideas have always been, well, what we'll do is we'll actually take the microbe and we'll kill it. And we'll give you a killed version of the microbe. So it's dead, it can't reproduce inside you. That's known as an inactivated vaccine. So again, that's going to be way safer, but it's not going to give you the same level of immune responses. It also may not get into the cytoplasm of cells really well, so it may not give you MHC class 1 presentation. So it might make, say, good antibodies, but no CD8 T cells, or something like that. So this is an inactivated vaccine. The flu vaccine, you've gotten, probably the polio vaccine you've gotten, many of the vaccines you get are inactivated. Those two vaccine types are the big classics. Those are the ones that most vaccines are. They're either live attenuated or inactivated. Researchers have come up with some other newer technologies to try to get around some of the issues with the other ones. One of the big sort of groups of those other technologies is sometimes what we can do is we can find out which part of the microbe is the key antigen. So we can say, well, we know actually you need an immune response against this antigen, which in the case of SARS-CoV-2 was spike. And so instead of giving you weak SARS-CoV-2 or dead SARS-CoV-2, we're just going to make some spike in the lab and give you spike. And then you'll make an immune response to spike, but you won't have any risk of getting sick because we're not giving you the whole virus. Does that make sense? So we're just going to make spike in the lab and give it to you. And so that there are different names for it depending on the different technologies for how that works. But usually we think about that as something called a subunit vaccine, where we're basically in the lab doing molecular biology to make the antigen and then giving it to you that way. So it's crazy, crazy safe. Your hepatitis B and your human papillomavirus are two vaccines that are subunit vaccines. So they're completely made in the lab against an antigen we know is a key antigen. They are crazy safe. But again, they may not get into the cytoplasm of the cell, so they're really good at making antibody responses, but not getting on MHC class 1 and making a good CD8 T cell response. So the question sort of has always been, how do you get the antigen into the cytoplasm so you can get the T cell response along with the antibody response? And the idea is, okay, so we know the spike is the antigen. Let's go backwards and think about the mRNA that encodes spike. So we're going to take the mRNA that makes the spike and put that mRNA into some of your cells. Now your cells will make the spike, and thus the spike will be in the cytoplasm. It'll get on MHC class 1. And so the idea is we basically have said, okay, we want spike, so let's get the gene that encodes spike and put that into cells. Doesn't go into every cells, it goes into a few cells in the arm where you inject it. Those few cells in the arm are going to present on MHC class 1, activate some T cells. There also will be some secreted spike that's made, so you get some B cells. And so now you actually get both arms of the immune system happening with mRNA vaccines. mRNA vaccines are amazing. There are so many great things I can say about mRNA vaccines. They are probably sort of the next generation of vaccines, both because we can make them quick, and they seem to be incredibly safe. And in grad school, I was actually working on a competitor vaccine. If you would ask me in grad school about mRNA vaccines, I would be like, that's the stupidest idea I ever heard. And now I'm like so proud that Drew Weissman and Callie Carico worked on it, even though everyone laughed at them. And they won the Nobel Prize and have basically brought us into a new era of vaccines. Yay. Again, I originally wanted 2 days for vaccines because I have so much I could say, but I can't. I've had an awesome semester with you guys. Thank you guys so much. I will send you some emails. There will be a review session on Sunday. I'll see you on Monday for the exam. Eat donuts.